Hi everyone, it's Jamie Batts, your instructor. We're going to cover chapter 13 right now, which is the lymphatic and immune system. This chapter can get pretty intense because of the immune chemistry stuff. So take it, as what it is and go slowly through the chapter, and hopefully you have enough time to, revisit this video and the PowerPoint, as well as work through the reading guide this week. Here's our learning objectives that are posted for you in the chapter folder. Let's start talking about the lymphatic system in general. So in general, the lymphatic system includes all of the cells, all of the tissues, and the organs that are involved in the defensive mechanisms that are involved in protecting our body. The hazards can be things like environmental hazards or internal threats. The specialized cells of our lymphatic system are called lymphocytes, and the vessels in the system are called the lymphatic vessels, or sometimes they're just abbreviated by lymphatics. The lymphoid tissues and organs are scattered throughout the body, all over the place, because you kind of want these little hubs of immune system defense all over the place. So let's talk about those lymphocytes. Those lymphocytes are the primary cells that are responding to invading pathogens, any abnormal body cells, as well as foreign proteins. So most of these lymphocytes are produced and stored within the lymph tissue or within the lymph organs. They are produced within the red bone marrow as well, and they're surrounded by lymph. Lymph is a liquid that somewhat resembles the constitution of the interstitial fluid. So it's kind of watery, salty. It's kind of that saltwater mix that surrounds cells. So here are all of the kind of parts of our lymphatic system. You have lymph nodes, you have some ducts, some cisterna chyli, which we'll talk about, tonsils, thymus, spleen, mucosa-associated lymphoid tissue or MALT, the appendix. All of these are lymphoid tissues. Let's talk about the lymphatic vessels specifically. They carry lymph from the peripheral tissue into the venous system. So we just finished learning about the circulatory system. All of the lymphatics they only move lymph in one direction, and it's moving that lymph back to the venous circulation. So it basically dumps back into the venous circulation right around the superior vena cava. So these vessels begin with lymphatic capillaries. The lymphatic capillaries are found within all the tissues and with all the organs. They're closely associated with blood capillaries. So where you have blood capillaries, you're also going to have a lymphoid capillary. The interstitial fluid flows into the lymphatic capillaries, and the fluid within inside those capillaries is called lymph. So again, that's why lymph resembles the interstitial fluid. It's basically the extra stuff that's sitting around after the blood capillaries have exchanged materials. The lymph sucks up that extra stuff. So here you see our capillary bed, venial side, and our lymphatics, which are just one direction. I think of these lymph vessels as kind of like little vacuum hoses sitting around the capillary beds, sucking up anything that wasn't reabsorbed, soaking up anything that wasn't taken in by the cells. It's taking up all of the extra stuff. It's pretty much what it does. So let's talk about these capillaries. It all begins at the capillary end. They're lined with endothelial cells, just like the inner linings of our blood vessels. They have an incomplete or sometimes missing basement membrane, so they're very loosely formed. They're not highly organized or specialized. They differ from blood capillaries because they originate as pockets instead of forming a continuous tube. So again, they're just kind of sitting there as kind of this vacuum hose that's sitting there, within your interstitial fluid, instead of the arteries that flow into the capillaries, which flow into the venules, which flow into the veins. It's just sitting there. They have very, very thin walls, and they're usually irregular because they have a very, very, very low pressure, because there's no pump system moving the lymph through. So the movement of lymph through these lymphatic vessels is one way. Those overlapping endothelial cells act like a one-way valve to allow the lymph in, kind of that vacuum suction motion as I was referring to earlier. It only permits the entry of fluids and solutes and not the return of those substances to the interstitial space or intracellular space. So here you see what I mean. They overlap, and once that fluid goes in, once those solutes go in, they cannot get back out, and they just continue flowing to to converge onto the larger lymphatic vessels. So lymph flows from the lymphatic capillaries into lymphatic vessels, and eventually it's coming back to the trunk of the body. The larger lymphatic vessels, which are closer to the trunk of our body, have valves, just like the valves of our veins. They're very close together. The vessels kind of bulge at each valve, and it kind of gives our lymphatics this arrangement that makes them look like a string of beads. Because we have that very low pressure in our lymphatics, the valves are basically preventing that backflow, just like the valves in our veins. So here you can see what I mean with the bulging, where those valves are, and again, closely linked to our circulatory system, running right alongside. So areas without a blood supply have no lymphatic capillaries. So something like the cornea of your eye, which is avascular, there are no blood capillaries there. Other areas without capillaries include our red bone marrow and the central nervous system, because again, there's no direct blood supply to our brain. It's all the CSF that's floating around. The lymphatics do not infiltrate that area. Sorry, I roll my sleeves up because it's very hot in this room when I close the door. This section, you can pause this part of the video, go back and answer these 3 questions. Let's keep moving. Let's talk about how extracellular fluid is circulated. So obviously our blood is confined into the vessels of our cardiovascular system, and the water and solutes will move from the plasma in the blood to the surrounding interstitial fluid. We talked about that in the last few chapters. The lymph contains the lymphatic vessels, is contained within the lymphatic vessels, and that's basically forming as the interstitial fluid is draining into lymphatic vessels. Again, that kind of vacuum cleaner hose that's sitting there sucking up that interstitial fluid. That's kind of what you should picture in your mind when you think of the lymphatic capillaries. So all of this, the blood circulation and the lymph circulation, is all about fluid homeostasis. It's the continuous recirculation of interstitial fluid because, as I mentioned before, where does all of this lymph go? It goes back into your venous system. It goes back to the superior vena cava, back into all of the blood circulation. So you're really not losing anything. This also helps to eliminate any local differences of nutrient levels or waste or toxins. So anything that's just sitting around accumulating in a tissue, the lymph is going to help suck it away, distribute it evenly throughout the body, or take it to the kidney so it can be eliminated. It also helps to maintain blood volume. All that interstitial fluid that's being sucked up by the lymph is going back again to the venous system, restoring the overall volume of blood. It can also serve to alert the immune system in case there's any infections in the peripheral tissues, and we'll talk about how that happens in the second part of this chapter. So here's the general circulation pathway. You have arteries carrying blood away from the heart. Then you have the solutes being reabsorbed or secreted at the capillary bed. Our lymphatics are soaking up anything extra that was not reabsorbed by the interstitial cells or by the venule cells, and taking all of that lymph, now as it's called, back into the venous system, which goes back to the heart and continues pumping around. So let's talk about how lymph is drained. Superficial and deep lymphatics all converge to form what we call larger vessels, and those larger vessels form the lymphatic trunks. The lymphatic trunks are just very large lymphatic vessels. Those lymphatic trunks empty into our collecting vessels. So you can think of the collecting vessels as kind of like the superior and inferior vena cava of the heart. This is where all of the vessels go. All of the lymphatic vessels are either leading to the thoracic duct or to the right lymphatic duct. The cisterna chyli, or cisterna chyli, it's pronounced chyli technically, is an expanded chamber at the base of the thoracic duct. What this does is it receives lymph from the bottom part of the abdomen, pelvis, and lower limbs, where we call the intestinal trunk. So our right lymphatic duct really just drains the right side of the head, the right arm, and the right side of the thoracic cavity. The thoracic duct drains everything else. So all the lymphatics from your feet, from your hips, from your left arm, from the left side of your head are all going back into the thoracic duct. The thoracic duct carries lymph, again, anything inferior to the diaphragm and the left side of the body. It empties into what we call the left subclavian vein, which goes back into the brachiocephalic vein, and which goes back into the superior vena cava. Same thing with right lymphatic duct. Right lymphatic duct is just collecting from the right side of the body, superior to the diaphragm, empties into the right subclavian vein. There you see them. They're very narrow and just kind of dump in right there. And then the subclavian turn into the brachiocephalic and then the superior vena cava. So let's talk about a condition called lymphedema. Lymphedema is caused by blocked lymphatic vessels. This is when the interstitial fluid will accumulate. This can cause very swollen or distended areas that are kind of swollen, extended, or uncomfortable. This usually affects the limbs, typically the lower limbs. It can affect other areas as well. The swelling could be permanent. Connective tissue will lose its elasticity, and therefore that area will just tend to be a little bit bigger than it's supposed to be because that connective tissue isn't rebounding. Stagnant interstitial fluids can accumulate toxins and other pathogens, so it's very important that our lymphatics be functioning and filtering properly. Local immune defenses would be overwhelmed if that interstitial fluid is just left sitting there and accumulating. Lymphedema, very uncomfortable. Doesn't have to be that extreme, obviously. But, you can pause the video here, go back and answer these 3 questions. Let's keep moving. Let's talk about lymphocytes. These are the active blood cells of our lymphatics and immune system. It's 20 to 40% of all of the white blood cells in our body are lymphocytes. So circulating lymphocytes are a very small percentage of the overall lymphocyte population. There's 3 classes. You have T cells, B cells, and NK cells, or natural killer cells. So before we talk about these types of cells, let's talk about antigens. All of these lymphocytes, T, B, and natural killer, are all sensitive to antigens. Remember antigens, those are those little surface proteins that allow us to tell blood type. Well, pathogens have them as well. All pathogens or parts of pathogens or parts of foreign substances make antigens. Antigens, for the most part, are proteins. Sometimes are lipids or polysaccharides. And ultimately, they're stimulating an immune response. So let's talk about the job of T cells. The majority of our circulating lymphocytes are T cells. There are a few major categories. You have cytotoxic T cells, or TC cells. These attack foreign cells. They are important in what we call cell-mediated immunity. So cell-to-cell combat, individualized combat. You were exposed to a cold virus before, and so you're immune to that cold virus now because you had it at one point in your life. So your T cells are going to remember that was there and attack. You have helper T cells. These stimulate the activation and function of T cells and B cells. And then you have suppressor T cells. These are there to actually inactivate or turn off your T cells and B cells. So our B cells are 10 to 15% of our circulating lymphocytes. They differentiate into plasma cells when they're stimulated. So B cells become activated and then they turn into plasma cells. Plasma cells are going to produce and secrete antibodies. They're responsible for an antibody-mediated immunity. Instead of the cell-on-cell combat with the B cells, with the T cells, I should say, the B cells are going to create these antibodies that float around in the blood and attack whatever they're specialized for. So some B cells will become memory B cells, and they will remain in your reserves and fight follow-up infections that have the same antigen. So this is how a vaccine would work. Exposing your body to the pathogen, allowing your body to create antibodies, and then when you're exposed to that pathogen in the future, your antibodies can attack. Let's talk about natural killer cells. This is a very small percentage of circulating lymphocytes. These cells will attack foreign cells, just like T cells and B cells, but they also attack the cells that have been associated with a virus or cells that are showing cancerous signs. So this is your body's way of monitoring itself through these natural killer cells. They continuously monitor our peripheral tissues. It's called immunological surveillance. So here you see the different classes of lymphocytes: T cells, B cells, natural killer cells, and then the 3 types of T cells, B cells differentiating into plasma cells, and then of course our natural killer cells are all pretty much the same. Let's talk about how lymphocytes are produced. It involves the red bone marrow, typically the thymus, as well as other peripheral lymphoid tissues. So the red bone marrow is the area responsible for maintaining a normal circulating lymphocyte production. We have hemocytoblasts, which you may remember from previous chapter on blood. These hemocytoblasts produce lymphoid stem cells. The lymphoid stem cells can migrate to the thymus. some of them will stay in the red bone marrow. The ones that stay in the red bone marrow will turn into B cells and natural killer cells. The cells that go into the thymus are going to be T cells, hence T for thymus. So the thymus, just like your brain, does not actually interact with the blood. There is a system of cells that are there to isolate it from the blood, and it's called the blood-thymus barrier. The thymic hormones are the hormones that stimulate T cell production, and T cells are produced and differentiate within that thymus, and then they re-enter the bloodstream. They can go back into the bone marrow or within peripheral tissues to mature and finalize their development. Here you see the overall process. It all starts in the red bone marrow with the lymphoid stem cells, which differentiated from the hemocytoblast. Some of them go to the thymus. They all come into T cells. Once they're T cells, they can go on back to the bone marrow to finish their development or develop in the peripheral tissues. The lymphoid tissue that stays in the bone marrow can differentiate into B cells or natural killer cells, again, finishing their development in the peripheral tissues for their specific job. Lymphocytes can reproduce. There are stem cells within the red bone marrow. B cells and T cells will kind of migrate to that area to produce daughter cells. It's very crucial for our immune response for those cells to divide successfully so that we have several of these copies of these important cells floating around our body. You can pause the video here, go back and review. Let's talk about specifics of our lymphoid tissue. I'm sitting on a pillow. Take it out. Okay, so lymphoid tissue is connective tissue that's dominated by lymphocytes. So the majority of the cells within the lymphoid tissue are either T cells, B cells, or natural killer cells. There's also lymphoid nodules within the lymphoid tissue. These are just little masses of lymphoid tissue. Lymphoid organs are different. Lymphoid organs are things like lymph nodes, the thymus, and the spleen. These are separated from surrounding tissues by a fibrous connective tissue layer. So let's talk about lymphatic nodules specifically. These are clusters of lymphoid tissues. They're found in the tracts that open to the external environment, like in the mouth, rectal area, reproductive organs, ears, eyes, all the openings. Deep to the epithelial, lining those passageways, and as a group, we call them MALT, mucosa-associated lymphoid tissue. So anywhere you have mucosa, like those openings, you're going to have lymphoid tissue there. It's meant to be one of your first defenses in your body, guarding that opening. The tonsils are one of them. There are lymphoid nodules in the walls of your pharynx. Pharynx is the fancy word for throat. You have the pharyngeal tonsils or adenoids. These are located on the posterior and superior wall of the nasopharynx. You have the palatine tonsils, which are located posterior and inferior along the boundary of the pharynx. And then you also have the lingual tonsils, which are covering the base of the tongue. Tonsillitis is when your tonsils are swollen due to some type of infection. This is just showing the locations of the general tonsils. And again, they're considered MALT. They're associated with that mucosal lining that opens the external environment. They're there for protection and surveying and making sure that invaders don't come in. Lymph nodes are different from the lymph tissue that we were just talking about. Lymph nodes are small organs that are shaped like a kidney bean. They basically function like a water filter. It's going to purify the lymph before it goes into the venous circulation, and it removes the majority of the antigens from the lymph. So this is the site of a lot of action when it comes to the immune system. Remember those lymphoid capillaries are just kind of sucking up all that extra interstitial fluid. Well, they don't just put all of it right back into the blood. That lymph is filtered, and the filtering stations are these lymph nodes. So your lymph nodes have what's called afferent and efferent lymphatics. The afferent lymphatics bring all of the lymph into lymph node. There are several of them leading into one lymph node. There is only one efferent lymphatic, which is carrying the lymph out of the lymph node. So you have all these passageways coming into lymph node and only one going out. The blood flow or the lymph flow through the lymph node is very, very, very slow, which allows all of the special cells that are in that lymph node to kind of check and monitor and destroy any pathogens, invaders, etc. So here you see what I mean. All of these afferent lymphatics Coming into lymph node, we have lymph node has its own blood supply to keep those cells happy, and then one efferent lymphatic here. So there's lots of B cells, plasma cells, dendritic cells, our little phagocytic cells that are in there gobbling things up. Let's talk about the thymus. The thymus produces hormones to help the development of T cells. Those are called thymosins. The size and secretory abilities are going to decline with age. So our immune systems, the versatility of our immune system declines with age because one of the reasons is because those thymosins are no longer secreted. In fact, the thymus starts to shrivel away with age as well. There is the thymus, kind of draped over the mediastinum area of our thoracic cavity. We also have the spleen. The spleen is the largest lymphoid tissue in the body. It has the same function for blood that the lymph nodes have for lymph. It's basically there to remove any abnormal cells through phagocytosis, recycle iron from the red blood cells, and initiate any immune response from the T cells and B cells. So anything that's going on with the blood is going to be monitored by the spleen. The spleen, if you ever touched a spleen, be a random thing to do, it kind of feels like a water balloon. It's very, very delicate structure. So you can pause the video here, go back and review these 3 questions. So let's talk about immunity. This is the second part of this chapter. Our immunity is our body's ability to resist infection and disease. So there's 2 forms of immunity. You have nonspecific or innate immunity, and then you have specific or adaptive immunity. Nonspecific or innate immunity does not distinguish one type of pathogen from another. So in other words, it basically is just a general form of defense. Doesn't matter what it is, your body's going to be protected against it. The response is going to be the same no matter what, and it's present as soon as you're born. It provides what we call a nonspecific defense. So let's talk about some of these nonspecific defenses. Physical barriers, like our skin, phagocytes, immunological surveillance, those natural killer cells. We all have them. They're all just there for the same reason. Interferons, which we'll talk about, the complement system, inflammatory response, as well as fever. These are all general nonspecific defenses. Then we have specific defenses, which are going to guard against particular pathogens. This is going to depend on the activity of specific lymphocytes. This is what we call specific resistance. This is when you get a vaccine to protect you against the flu, or when you had a certain cold before, and so you're not going to get it again. It's because you have a specific defense against that particular pathogen. So again, some examples here. Physical barriers like our skin, you have phagocytes like monocytes and macrophages, as we've talked about before, our immunological surveillance performed by our natural killer cells, interferons, which are just chemicals that are released to destroy cells, the complement system, again, it's a cascade pathway to destroy cells, inflammatory response, and fever, all there as a protection mechanism. So let's talk about the integumentary system in general. Obviously, it's providing a major physical barrier. There's glands, hair, and epithelial coverings. They all have protective immunological defense mechanisms built in. The glands are secreting destructive enzymes. They're secreting lysozymes, which have bactericidal elements to them. They have antibacterial properties in all of our body secretions, from our tear ducts to our mucus to our sweat and oil. All of it contain bacterial enzymes, antibacterial enzymes. Our hair is also there to protect against mechanical abrasion, as well as the sebaceous glands, which are associated with our hair for the oil secretions. And our epithelial covering, which has several layers of keratinized cells to help prevent dehydration, as well as prevent anything from coming into the body, kind of has interlocking cells that go together to prevent invaders from coming in. So this is just looking at the physical boundaries of our integumentary system. Those epithelial boundaries also line the tracts of our body, like the digestive, respiratory, urinary, and reproductive tracts, providing another line of physical barrier. The secretions, again, have antibiotic properties. They have special enzymes called lysozymes in it. Our stomach has acid to help destroy pathogens. There's also mucus in the respiratory system to destroy pathogens. The urine is going to flush out the urinary tract or urinary passageways, and there's also glandular secretions of the reproductive tract to help protect those areas. Molt, again, our mucosal-associated lymphoid tissue provides that nonspecific defense in those areas as well. And there you see them, nonspecific defense barriers. Let's talk about phagocytes. Phagocytes are our first major line of cellular defense. These are found in all of our peripheral tissues. They're there to remove cellular debris, respond to invasion of foreign compounds or pathogens. They can also attack or remove any microorganisms before the lymphocytes even detect a presence. So these are like the little Pac-Men floating around your blood, gobbling things up, gobbling up the bacteria or pathogens or waste products that don't belong there. They all have different types of target threats that they're looking for, but they all function the same way. They all eat phagocytes. There they are, kind of eating the bacteria, these little rod-like things. And this phagocyte's just engulfing those little rods. There are several types of phagocytes in our body. I mentioned some of them before. Neutrophils are the major one. This is most abundant. This is mobile. It's very fast acting. Remember, these cells use that, that process of chemotaxis where they just kind of sniff out the invader. They smell something that doesn't belong. Eosinophils or eosinophils, these are much less abundant, but they phagocyze foreign particles and pathogens that have been coated with antibodies, so anything that's been attacked already by the B cells. And monocyte macrophage system involves the macrophages, which were made from monocytes, fixed macrophages, as well as free macrophages throughout the body. There you see them. Let's talk about other characteristics of phagocytes. They can leave the capillaries by squeezing through the endothelial cells. It's called diapetes. You don't need to know that. They may be attracted or repelled by chemicals. That's chemotaxis. We mentioned that already. The receptors on the plasma membrane can bind to the surface of the target, and then after attaching, the phagocyte either destroys the target or promotes the destruction through other specific defenses like calling in or alerting other B cells or T cells. You can pause the video here, go back and answer these 3 questions. Let's talk about inflammation. Inflammation is another nonspecific defense. It's going to occur in the same way no matter what the pathogen is. It's a localized tissue response to some type of injury. It's caused by swelling, or it produces swelling, redness, heat, and pain. It's caused by a number of stimuli. The stimuli are meant to kill cells or damage connective tissue, fiber, injured tissues. Ultimately, after fever has occurred, the chemical composition of the interstitial fluid may be altered. Prostaglandins, protein, potassium ions, etc., foreign proteins or pathogens can all come into that area. So all of those things are triggering the inflammatory process. Let's look at the inflammatory process. So you have some type of damage, and then the mast cells are activated. Mast cells release histamine and heparin. Histamine and heparin are going to dilate the blood vessels and help clot the blood. The phagocytes are attracted to the area through chemotaxis, and they're basically going to alert the rest of our body's defense. Possibly even specific defenses may be activated in this case, or fibroblasts may be stimulated to rebuild or secrete collagen fibers, etc. Ultimately, the tissue would be repaired and the scar would form. So, fever is our last nonspecific defense. This is a rise in core body temperature caused by pyrogens. Pyrogens are basically circulating proteins that reset the thermostat within the hypothalamus. Can be very beneficial within limits. It will inhibit viruses and bacteria from spreading, increases our body metabolism to mobilize defense mechanisms, and help accelerate the repair process. So overall summary of nonspecific defenses: physical barriers, phagocytes, immunological surveillance, interferons, which are released by lymphocytes and basically trigger antiviral proteins. We didn't have a whole little section on that before, but ultimately they stimulate macrophages and natural killer cells. The complement system, which is just a group of circulating antibodies or proteins that are going to help antibodies destroy pathogens. Inflammation and fever we've talked about. So there they all are on this nice pretty picture. You can pause the video here, go back and review. Let's talk about specific immunity. Specific immunity protects against specific pathogens. So it can be a coordinated activity of T cells and B cells that give us a specific defense. T cells are primarily responsible for that cell-mediated immunity where they defend abnormal cells or defend against abnormal cells and pathogens inside cells. B cells provide antibody immunity, which are body system wide. They're looking, through all the body tissues and all the body fluids. Specific immunity can be active or passive. Active immunity is when your body actively produces antibodies in response to the antigen. Passive is when they receive antibodies from another source. They can either be actively naturally acquired or artificially active acquired, or passive naturally acquired or artificially passively acquired. So in other words, our active and passive immunity can either be naturally acquired or artificially acquired. Let's look at some examples here. In our specific immunity, which we're talking about now, you have active immunity, which is, again, where our body's naturally or where our body is specifically forming a particular antibody to a particular antigen. It can be naturally acquired or artificially acquired. Naturally acquired would be where you are exposed to a antigen and you develop the antibodies for it. So last year you had the cold, and this year that same cold virus comes around, and now you're naturally immune to it because you acquired through natural sources that particular antigen. You made the antibodies actively, so it's naturally actively acquired immunity. Artificially induced active immunity would be like when you get a vaccine. So you're getting a flu vaccine maybe this fall. They're giving you something to induce your antibodies to be made. You're still actively making those antibodies, but it's through a artificial process. It's not a naturally acquired process. Passive immunity is where you are passing antibodies from one source to another. They can be naturally acquired, like through the placenta or through breast milk. Or they can be artificially acquired, like in something like a rabies vaccine, where they're injecting you with antibodies. They're just straight up giving you the antibodies. Your body's not going to have time to make them, so they're giving them to you. And so those are the ways your body develops specific defenses. Let's talk about some properties of specific immunity. Obviously, specificity is the first one. Each T cell or B cell has special receptors to bind to one specific antigen. It's going to ignore all of the others, kind of like an enzyme and substrate. They fit together like a lock and a key. The response of an activated T cell or B cell is so specific that only that one antigen is it going to respond to, nothing else. Our specific immunity is also versatile. Millions of different lymphocyte populations, each is sensitive to a different antigen. So when it's activated, the appropriate lymphocyte divides, producing more of that same lymphocyte so that you can carry out this specialized attack. All cells that are produced will be like that, will be clones of the one that is needed. Our specific immunity also has immunological memory, meaning activated lymphocytes will actually produce two groups of cells. They'll produce attacker cells, which will go off and attack, but they'll also produce inactive cells that serve as a memory of what that active cell is supposed to be. We call them memory cells because they remember the antigens, and they will launch a faster, quicker, more efficient attack when they're exposed to those antigens in the future. Our specific defenses also have tolerance. Our immune responses will actually ignore the cells that are supposed to be there, and they will specifically target all of the foreign or abnormal non-self cells. This can develop over time, over the exposure to a particular antigen as well. So if your body's under chronic attack from a particular pathogen, your body could get a tolerance immune system to it. So let's talk about some triggers for our specific defenses. Exposure to an antigen, a bacteria comes into your body. It's going to first activate phagocytes. Phagocytes are going to stimulate a cell-mediated defense involving T cells. They're also going to trigger an antibody-mediated defense. The first step in the whole process is called antigen presentation. It's basically here you see the whole process. Specific defenses, virus or bacteria comes in. At the cell-mediated level, our phagocytes are activated, our T cells are activated, and our antibody-mediated response, our activated B cells are going to start producing antibodies. There's a feedback mechanism between the two. So let's talk about antigen-presenting cells. These are special cells like monocytes or macrophages or dendritic cells, all cells of our immune system. Don't get the word dendritic cells associated with our nervous system. Dendritic cells are not neurons. They don't have dendrites. They resemble those neurons, but they're specific immune cells. These special cells will engulf a pathogen, and then they will basically digest that pathogen and produce some of those antigens on their own cell surface, these little fragments. And that is going to basically signal that to all of the other cells that an invader is there. It inactivates and takes the threat away, but it's basically sending up a little flare saying, oh my gosh, we're being attacked by this particular antigen. So here is an antigen-presenting cell. You have this bacteria that's being engulfed by this phagocytic cell. The phagocytic cell is going to digest and break down that bacteria, and it's going to take those little antigens and present them on their own cell membrane. So, our overall defense against bacterial pathogens is initiated when those macrophages are activated when they present that antigen. There are also defenses against viruses. Viruses involve a direct contact with virus-infected cells. The antigen presentation by the antigen-presenting cells, those APCs that we just mentioned, as well as interferon. So it's a little extra step with virus-infected cells. So with bacteria, again, those macrophages are activated, they engulf, they throw up that little flare with the antigen on it, and then that's what's called antigen activation or presentation, and that's where the memory T cells are created, and then the cytotoxic T cells or B cells will be created to destroy that pathogen. In viruses, again, viruses will infect a tissue or infect an antigen-presenting cell. They'll actually infect the macrophages. And those macrophages will do the antigen presentation, put up their little flare, and then helper T cells and B cells will come out and destroy the virus. If they infect tissue cells, those interferons are going to be released. Interferons are basically going to spread viral resistance to the surrounding area and help the spread of the viral particles help spread the viral particles going everywhere. Those tissue cells are going to throw up their antigen on their plasma membrane, just like the macrophages did. And overall, T cells and B cells will come in and destroy that cell also. All infected cells by viruses are destroyed. So memory cells play an important role. They don't have an initial role, but they have a role later on. They will dramatically reduce the response time when that antigen is presented again. Upon secondary exposure, they will differentiate either into helper T cells or into cytotoxic T cells or into memory B cells. It's a very rapid and effective response. You can pause the video here and go back and review. Let's talk about antibodies. Antibodies are polypeptide chains, so they're protein chains. They're made of heavy chains and light chains, and each chain has a constant and a variable segment. So the constant segments form the base of the molecule, and the variable segments will vary from antigen or antibody to antibody. So the free tips of the two variable segments are called the antigen binding sites. They will interact with an antigen that's similar to the way an enzyme acts with a substrate. So they fit together like a lock and a key. The differences in the variable segment structure will affect the shape of the binding site. So antibodies specific for different antigens. So they are all very, very specific to each other. Variation can occur during production or division or differentiation, so they have to be made precisely in order to work. Here you see what I mean. Here's the light chain. This is the variable segment. This is the other variable segment. These are the binding sites for the antigen to kind of clip on to the antibody, or vice versa. The antibody really clips on to the antigen. So the antibody binds to an antigen to form what we call an antibody-antigen complex. The binding sites between the antibody and antigenic determinant sites are very specific portions of the antigen surface, and bacteria can have many, many, many of them, millions of them. So antigens come in complete or partial forms. A complete antigen would have two antigenic determinant sites, and otherwise it would be able to bond in two spots to the antigen. A partial antigen is sometimes called a hapten, and they don't ordinarily activate B cells. So they can attach to what we call a carrier molecule, and then they will function as a complete antigen. And the antibodies will only attack the hapten and the carrier molecule if they're both together. If the carrier molecule is normally found in the tissues, the antibodies may actually attack normal cells. This would be why our body would be like allergic to something like penicillin, because it attaches to a carrier molecule in some cases, and then our body will recognize and attack it. So here is the basic idea. Antigenic determinant sites are these two binding sites for the antigen and antibody. They fit together in a complete and an incomplete or partial. They have to fit together with a carrier molecule and a partial antigen. Hence there you see them binding onto the surface of some type of invader attacking it. Antibodies are all different. They have classes of, they're based on their structure and function. They're also known as immunoglobulins or Igs. So there's IgG, IgE, IgD, IgM, and IgA. IgG, this is our general defense. This is the majority of our antibodies are classified as IgG. This is 80% of them. This is the bacteria. This is the viruses. This is the majority of your immune responses. IgE is going to attach to our basophils and mast cells, helping with inflammation, speed up the process of inflammation. IgD is on the surface of B cells, and these play a role in sensitizing B cells to produce more antibodies. IgM is encountered when we are dealing with blood types. So this is the antibody that's produced in a transfusion reaction. And IgA is in gland secretions. This is the antibody that's in our tears, our sweat, our oil, etc. So let's talk about the first time you've been exposed to an antigen. It's called a primary response. This is when you're initially exposed to some foreign body. It takes time to develop resistance to it. The antigens have to activate the B cells, and the B cells will then differentiate into plasma cells, and then the plasma cells will secrete the antibodies. There's something called an antibody titer, which is going to peak 1 to 2 weeks after exposure, and then it will decline if there's no more exposures. A secondary response is triggered when that antigen is encountered again. So it's typically more extensive and can last longer than the primary. So the antibody titer increases rapidly and to a much higher level, much quicker. So it's almost an immediate response by those memory B cells. It appears even if the second exposure is years and years or decades after the first exposure. The memory cells can survive 20 or more years. So here's what we mean. After you've first been exposed to West Nile virus, some type of virus or whatever, your IgM is going to take time to grow in high concentrations to provide defense. This is when you start to get sick. You're starting to feel, that you're not right. Maybe you are taking a day or two off work. A week into being exposed to that antigen, that pathogen is starting to really take over your body. It's right about when your IgM and your IgG antibodies are finally taking over and taking control back of the body. And they always say about 2 weeks, right? And so after about 2 weeks, that's when that antigen is usually wiped out of the body. If you get exposed to it again, it's almost immediate. It's not even a week long before you reach IgM peak levels. And look at the IgG. It reaches an even higher level, and it stays elevated for weeks and weeks after. So you can pause the video here, go back and answer these 3 questions. Let's talk about how antibodies work. They can neutralize, they can prevent pathogen adhesion, they can activate the complement, which we talked a little bit about. They can do something called opsonization. They can attract phagocytes, they can stimulate inflammation, or they can do agglutination, which is something we talked about in blood types. Let's talk about neutralization. This is when bacteria and viruses have to bind to the plasma membrane before they can enter our body cells and infect them. So the antibodies are actually going to attach to the binding sites of those toxic cells, basically neutralizing them. Once something's already bound to them, they can't go and bind themselves to something else. So that's what neutralization is. There's no more binding sites available. You can prevent pathogenic adhesion. Basically, antibodies that are part of our saliva, mucus, tears, etc., basically are covered in antibodies to create a barrier. So this makes it difficult for pathogens to adhere to our body's surface. Binding an antigen changes the antibody's shape, the molecular shape. So complement proteins are proteins that are going to bind to the antigen and change that shape so that they can no longer bind and are inactivated. Opsonization is a coating of antibodies and complement, those extra proteins, again, on the surface of bacteria to make them less slick so that phagocytes can attach more easily. So it's kind of a team effort to get the bacteria to be a little bit more sticky so that phagocytes can grab on and they won't slip away. Phagocytes can be attracted, so neutrophils, eosinophils, macrophages are all attracted when the antigen is coated with antibodies. And inflammation can be stimulated when the antibodies are releasing heparin and histamine. That's, of course, coming from basophils and mast cells, promoting that overall inflammatory response. And agglutination is when large number of antigens together create what's called an immune complex. And this is going to form that agglutination process that we saw. It's basically clumping of the antibodies around the antigen. So there's agglutination. And then all of the antigens tend to clump together also. So all of the bacteria start to clump together. You can pause the video here, go back and answer these 3 questions. Let's talk about how our antibodies can overreact. Something like an allergy is an inappropriate or excessive response to an antigen. This could be a sudden increase in activity or antibody titers, the concentration of those antibodies. Neutrophils and cytotoxic T cells may start destroying normal cells instead of just the antigens they're designed to attack. The antigen-antibody complex can trigger inflammation, or antigens alone can trigger an allergic reaction. So, an initial response to an allergen is something called sensitization. It produces large quantities of IgE antibodies, and you have immediate hypersensitivity. This is a rapid, severe response to an antigen. This would be like hay fever, right? You go outside sometimes in the spring and you immediately start sneezing, the nasal membrane becomes inflamed. Hypersensitivity reaction can be limited to typically to just the body surface. Sometimes it can be systemic where you have an overall rapid reaction. The allergen can trigger that systemic response, as I mentioned, that would be anaphylaxis. Anaphylaxis is when that antigen starts circulating in the body and starts stimulating histamine from mast cells throughout the entire body. So this is peripheral vasodilation to the extreme drop in blood pressure because everything is dilating, and this could lead to anaphylactic shock. So here you see the overall reaction, these allergens coming in, sensitizing and activating B cells, activating macrophages, T cells are activated, ultimately making lots of IgE antibodies, sensitizing the mast cells and basophils, and eventually those histamine and other chemicals can cause pain after the second exposure. It can just keep getting worse and worse. You can pause the video here, go back and review. Let's talk about some disorders of the immune system. They can be basically a immune system gone wrong in most cases. Autoimmune diseases, which is basically where your body's attacking itself. It does not have the ability to recognize self from non-self, so certain parts of the body are being attacked. There's also a graft rejection, and allergies are another response of an immune disorder. There can also be an inadequate immune response. This would be like an immunodeficiency diseases like HIV or age-related reductions in immune activity. Let's talk about autoimmune disorders. In autoimmune disorders, the body is attacking itself. There's a malfunction of the self-antigen recognition system, and our cells start to produce autoantibodies. This is about 5% of the adults in North America and Europe, and they're usually caused when proteins are kind of similar in structure to one another. So in other ways, A protein that's very closely associated with measles, Epstein-Barr, and influenza has the same amino acid as the myelin proteins around our nerve cells. So the antibodies that are meant for measles, Epstein-Barr, or flu can readjust themselves and then attack your myelin, right? That would be the case of MS, multiple sclerosis. Possibly, it's a theory. Antibodies targeting those pathogens can also attack the myelin sheaths, and like I said, it's a mechanism for possibly multiple sclerosis. There's also thyroiditis. This is the inflammation of the thyroid gland. Rheumatoid arthritis is when the autoantibodies are attacking the connective tissue at your joints, causing inflammation and widespread destruction at your joints. Type 1 diabetes is where your autoantibodies attack your insulin-producing cells of your pancreas. There's rheumatoid arthritis, very painful. The graft rejection would occur after an organ transplant. So the recipient's T cells are activated when that donated tissue becomes attacked and destroyed by the foreign cells. So the transplant success can be improved by something called immunosuppression. This would be reducing the sensitivity of our immune system. Cyclosporin A, or CSA, will suppress the immune response by inhibiting those T helper cells, but not affecting the suppressor T cells. So your overall immune system isn't totally knocked out in that case. The immunodeficiency diseases can result from an embryological development issue with the lymphoid tissue or lymphoid organs, some type of virus that suppresses our immune function, or treatment or exposure to immunosuppressive agents. There's also, as I mentioned, age-related immune degradation. That's the word I guess I was looking for. So let's talk about AIDS. Acquired immunodeficiency syndrome. It's the most common immunodeficiency disease. It's caused by the human immunodeficiency virus, HIV. The virus is going to bind to special CD4 proteins and infect our helper T cells. So those infected cells will actually synthesize and release new proteins, and they will be destroyed by the virus and the immune defenses. So it basically impairs our cell-mediated and antibody-mediated responses. The suppressor T cells are not affected by the virus, but what does generally happen with people that have suppressed immune systems, in this case, they would be more susceptible to opportunistic infection. So people that have AIDS typically don't get sick from the AIDS. They're getting sick from the cold or the flu that you or I can fight off very, very easily. So HIV is spread through contact of body fluids, as we all know now, but back in the 80s, they didn't know how it was spread. So it's a relatively new disorder when we are looking at viruses that have had such a wide impact on our lives. It affects 33 million people worldwide and causes 2 million deaths per year. There's HIV coming out of an infected T cell. Other age-related changes or reductions in immune activity. As with everything, it all slows down, gets smaller, gets weaker. The immune system is less effective with age. The thymus shrinks. The thymic hormones are decreasing, so your T cell production is going to decline. Increased susceptibility to viral and bacterial infections because your T cells are less responsive. There's a fewer number of cytotoxic T cells. The number of helper T cells is reduced, so that your B cells are less responsive. Vaccinations are strongly recommended in the elderly, and an increased incidence of cancer will also cause a decline in immune response. There's this guy getting checked out. And last 3 questions of our chapter. Let me know what questions you have. Immunity can get very, very tricky, and I tried to simplify it as much as I could for you in this chapter. Your book does a good job of keeping it simple, but it is kind of overwhelming because there's a lot of cellular chemistry involved. So just focus on the big picture stuff and don't get bogged down with the details of the immune response because there's a lot to it. Let me know what questions you have and have an awesome night.